All right, well, that was one important thing about transcription. Now, the next thing we want to see is the idea of this, the foundation of transcription in eukaryotes. You have, first of all, you have this sequence, the promoter. Inside the promoter, in most, but not all, the vast majority, but not all, there is something called the Tata box. It has the sequence TA in one of the two strands of DNA. And that is highly conserved. It's found in the vast majority of promoters, especially. We're focusing on the RNA polymerase II promoters, in other words, the ones that make messenger RNAs. Promoters for polymerase 1 and 3 are similar, but do have some difference. Recall the polymerase is a little bit different for each one of them. But most promoters for all of them do have Tata boxes. For the polymerase 2 and the polymerase 1 as well. Okay, now this promoter sequences tend to be about, say, 30 base pairs long or so. So they're fairly short sequences. And they tend to be located, maybe about 20 or 30 or 40 bases from the actual gene. Okay, so here's our promoter. Now the promoter in prokaryotes was the binding site of the RNA polymerase enzyme. In the case of eukaryotes, we're going to have, instead of just the polymerase, we have a number of different proteins that assemble together, bind the polymerase, position it just right, and other proteins activate the polymerase, because it turns out that the RNA polymerases in eukaryotes are generally inactive, and then they have to be phosphorylated in certain locations in order to fire them up. So they're regulated proteins. So here again, that's another control. An inherently inactive polymerase is another control to keep the wrong genes from being transcribed. You have to switch the polymerase on in order to get transcription to take place. So that's another kind of thing. So you just put a garden variety polymerase, stick it on the promoter. Unless you activate it, it's going to sit there staring at you. So that's another level of control to keep the wrong genes from being transcribed. Okay, so we have a number of proteins that are going to bind to the promoter. And these form what's known as the basal complex. The basal complex consists of the RNA polymerase and several what are called basal or general transcription factors. And each of these basal transcription factors are going to play important roles in things like positioning the polymerase, activating the polymerase, doing things that are necessary for transcription to take place. The polymerase cannot do all that stuff by itself. It needs all these guys to help the process along, to do critical things. Now here again, when we look at prokaryotes, the polymerase basically, if it sticks to the appropriate promoter, it's ready to roll and it does what it needs to do. Here we got to have a lot of help. That's our basal transcription factors. And there's several of these things. Many of them are multi-subunit proteins in their own right. So when we put the whole basal complex together, it's a pretty complex assembly of proteins. Now, the basal complex is going to be the minimum requirement for transcription. Without the basal complex, you will not get transcription at all. You have to put all of that stuff together, every single one of those proteins involved, for transcription to take place. Yet, despite that, even the basal complex alone, many times it does not transcribe unless you have other specific transcription factors to help it along. So here again, looking at eukaryotes, we have multiple ways of keeping a gene switched off. In other words, not being transcribed. And to transcribe a gene, we have to do a whole bunch of things to it. We have to push the nucleosomes out of the way so that we have our sequences exposed. We have to get this large basal complex together and keep it down on the promoter long enough so we can fire off the polymerase. And most of the time we need additional transcription factors to help stabilize that stabilize that basal complex so it can actually do its job. So once again, off, off, and off. And to switch transcription on, we've got to do multiple things in order to switch it on. And that here again is very important in development. If you're a single-celled bacteria, for instance, most of the time, if you accidentally transcribe a gene you really don't need to transcribe right now, you know, maybe it codes for an enzyme that can digest a food source that isn't there. Okay, what do you do, folks? You switch on a gene, and what's going to happen? You wasted a few amino acids, wasted some ATP, you wasted a little bit of energy and stuff, but it's not really going to hurt you all that bad. But when you look at a multicellular organism that undergoes development, if you switch the wrong gene on, say a gene coding for a transcription factor that activates a whole developmental pathway, you're in serious trouble. You switch the wrong gene on. For instance, there's a gene that first identified in fruit flies called the eyeless gene because lack of it meant you had no eyes. You developed normally except your eyes didn't form. In mammals, it's called PAX6. It does the same thing. It generates the program to develop an eye. Now imagine if you happen to switch that gene on in the wrong place. You could have eyes in the back of your butt, bro. In fact, experimentally, they did something like that with fruit flies, and sure enough, when they switched the eyeless gene on in the leg of a fruit fly, what was going to be the leg of the fruit fly, they actually got compound eyes forming on the knee and signs of innervation. Whether they could actually see through them or not was hard to say. Nobody could really tell. But, you know, that gives you an idea. What would happen if you switch the wrong gene on, a single gene, especially if it's like a transcription factor or a major kinase or something, switch the wrong gene on in the wrong cell, you can get a developmental disaster. So we want to make absolutely certain that before we transcribe something, we want to make darn sure that that gene, do you really want to do this? You know, a little warning on the screen. You are about to transcribe this gene. Do you really want to do this? Yes, no, you know. And because they have to be careful. So keeping genes off and having to do multiple steps for transcription to take place is a good safety valve. It keeps mistranscription from occurring, at least much of the time, and it also allows you to make sure that only certain genes you want in that particular cell type are going to be transcribed and that you're not going to have too many accidents occurring elsewhere. Okay, well, so we have the basal, this is the basal complex, is the minimal complex for transcription. The other thing is that we can regulate the basal complex with other transcription factors. And we're going to see these very specific transcription factors that ramp up or ramp down transcription. What they ultimately do is they do something with the basal complex. They may stabilize it, increasing transcription rates. They may push components off of it, causing the whole thing to collapse and stopping transcription, or any of a number of things. So I'll put TFs for transcription factors. Okay, so another point here. Other transcription factors will interact in one way or another with the basal complex, and that affects transcription. So in effect, regulating transcription is largely dependent on doing something with the basal complex. That's where all the action occurs. Now, before we go on, let's kind of take a look at the key players of the basal complex. There's several different proteins. We'll just mention them briefly and see what their functions are. And once again, we're going to focus on the polymerase II. There are differences in polymerase I promoters and things like that. The basal complexes are quite similar for the polymerase I, polymerase II, polymerase III, but there are some different proteins for each one of them. So let's focus on the polymerase II promoters. Okay, first thing, there's a protein called the Tata binding protein. The Tata binding protein, with a name like that, you can assume it binds to the Tata box, and yes, it does. Okay, the Tata binding protein, or TBP, is the cornerstone of the whole basal complex. It's the first thing that goes in, and it's going to be the foundation of the assembly of the rest of the basal complex. Push the TBP off, and everything collapses. So that's probably one of the most important one of these basal transcription factors. That starts the whole assembly process off. Now, it can also be associated with other proteins, and we'll deal with these guys later. When you assemble these other proteins, we make a much larger complex called TF, that's Transcription Factor 2D. The 2 represents for polymerase II. Okay. So the Tata binding protein assembles with other proteins to make a much larger thing, our TF2D. We'll see what that guy does later on, but yes?
What is that C word?
C word, cornerstone. Okay, so it's cornerstone or foundation would be another good term for it. So yeah, you know, if you build a structure, you don't have a foundation, what happens? It collapses. If you got a building, you pull it, you wreck the foundation, what happens? It collapses. TBP is like that. That's where everything starts. You don't have that, you got nothing. Okay. Other characters. TF, Transcription Factor 2A. It stabilizes the TBP. In other words, it helps to hold the thing down. Otherwise, the TBP can pop off spontaneously. This pins it down to the Tata box where we want it to. Coming in is going to be our TF2B. To B or not to be. Okay. What this does is this is going to be what actually binds the polymerase. In other words, when we're looking at prokaryotes, when we're looking at bacteria, the polymerase, its major interaction was binding directly to the promoter. So you had a DNA and protein interaction here. In this case, the polymerase really doesn't bind much to the DNA at all. It doesn't bind very tightly to DNA at all. Instead, the main thing that holds the polymerase down is another protein. And here's our TF2B is the one that does it. It grabs the polymerase, holds on to it, and kind of settles it over the right DNA sequence. And, closely related, is another one, the TF2F. What it does is it helps to not only stabilize the polymerase, it helps to position the polymerase so that the right parts of it are over the right DNA sequences. Because here again, now think of this. You want to start transcribing. We have a certain site the transcription start site. It's just a few base pairs away from the gene. If you start transcribing here instead, you're going to get a bunch of sequence you might not want. That could lead to problems when you finally do the processing of the RNA. It could lead to the to an RNA that's got some kind of defect in it. Or if you ended up starting here, what's going to happen? You're going to miss part of a coding sequence. And that means the messenger RNA is going to be missing something. Now, that can be very bad. Recall what happens with that signaling protein, that G protein involved with signaling pathways called RAS. If you miss the front part of RAS, mutation, common set of mutations removes that front part. That contains the GTPase site. In other words, you miss the front part of RAS. Once you switch it on, it can't hydrolyze GTP, so it continues signaling permanently. Bad idea. It's found in a lot of cancers, those kinds of RAS mutations. So you want to make sure that the polymerase is put exactly in place to the base. If you don't, you're going to get yourself problems. So our old friend TF2F is the one that does that. And we got a couple others here. Transcription factor 2E, TF2E. Okay, one of the things it does is it's a kinase. It phosphorylates proteins. And specifically, its target is our last of these basal proteins here. Oops, sorry folks. Okay. Our last member of the basal complex, TF, or transcription factor 2H. 2H is an interesting protein. It's what's called a helicase. I'll describe a helicase in just a moment. And it also is a kinase. And when the kinase part of this protein is activated, what it does is it phosphorylates a portion of polymerase 2. And that phosphorylation activates the polymerase. Recall we said that the polymerase 2 is inactive until it's switched on. And switching it on involves phosphorylation in a particular region of the protein, and that switches on its activity, and then starts to roll after that. Yes?
The TF2E under kinase.
Okay, it says helps activate a helicase, and specifically phosphorylates and activates this guy, TF2H. Now, what is a helicase? Okay, well, probably best done if I do a little demonstration here. Okay, here's a double strand here. Now, I'm going to twist this guy around a little bit, right? These two cords here. DNA, the double helix. So this is a model of a double helix, right? Okay, now when you're going to start transcribing a gene, what you have to do, of course, is you have to break the hydrogen bonds between two strands of DNA, separate the strands, and then move along like so. Okay, now, if I do this, I move along, you notice I'm tightening up the coils on this end as I push further and further up. And if I continue to do this, I'm not going to climb up to do this. If I continue to do this, I'm going to twist more and more. This is going to be super twisted up here. And that adds resistance. And eventually the resistance will get to the point, this super coiling of DNA generates so much resistance that the polymerase can't push any further. And if I were to do that with my hand here and stand up on a chair, I could push a little bit further, and finally I would not be able to push any further at all, as much as I've tried. Okay, so that would be a problem. In other words, as you're pushing along the DNA and it's getting more and more tightly coiled up ahead of you, it's eventually going to get to the point where you can't move any further. Bad idea. So what we got to do is relieve that stress. And that's what helicases do. what a helicase does what a helicase does is when it starts getting tightly coiled, we cut the DNA on one strand, and that allows it to uncoil. And then we glue it back together again, restoring the original coil. So helicases they relieve that supercoiling we get with the DNA if you just continue to push along the double helix. They cut one strand, allow it to kind of unwind a bit, and they glue it back together again with the original coiling. And so the helicases actually move a little bit ahead of the polymerase to keep on doing this. The polymerase never runs into too much resistance. So we got a lot of complex functions in this base of complex. Now I'll probably just kind of like do a little bit of assembly here on this right over here. Is it okay if I kind of erase our players a little bit? We kind of mentioned, yes?
Quick question. You said TF2F, what position, what does it stabilize?
Okay. It helps to stabilize the polymerase, and it also helps the position. And the other one, the TF2E, or rather the TF2B contributes to that too, because the 2B binds it, helps to stabilize, but the polymerase is a huge enzyme complex. It can pop off real easily. So now 2F comes in and helps it to stick even more, and also make sure it gets into the right position. Okay? All right, well, let me get, okay, we'll get rid of our little group of players here for a base of complex. And once again, all these different proteins are the minimum requirement for transcription. Now you can imagine if we were to push any one of those basal transcription factors off, that's going to stop transcription. If you push the TBP off, the whole basal complex is going to fall apart on you. Okay, so the first thing that's going to happen is TBP binds. Binds to our Tata box. It's interesting, they do have a structure that, it turns out TBP, one thing that it does is it's actually kind of a V-shaped protein, and it binds the DNA and actually bends the DNA at a very sharp angle. One thing that does is it puts stress on the double helix and it helps make it easier to separate the two strands. It's I used to call, quote, a torture rack for DNA. You know, you look at it, it's like the Spanish Inquisition got to that poor promoter site, you know, because it really bends it pretty extremely. Okay, so that's our first thing. That's our cornerstone here. And 2A comes in and helps stabilize the TBP, because otherwise it can fall off. This helps prevent it from falling off. Now, in comes 2B, to be or not to be. It binds the Tata binding protein, but it's going to be our binding site for the polymerase. And the polymerase actually comes in as a complex. Here's the polymerase. Actually, it's much larger than this in real life. And we have it attached to it is going to be 2F. So now we've got the polymerase helps bind to, okay, maybe a little bit more like this. The polymerase is going to bind to TF2B. It's also going to bind to 2F, and both of them together help stabilize the thing. I'll stretch it out so it really does touch the thing. Both of these things are going to stabilize and position the polymerase. So it's right where we want it, and the appropriate part of the polymerase is right over our transcription start site. That's what we really want here. Okay, and then comes in, it sticks to the polymerase and to 2F. That's going to be our kinase that phosphorylates the final thing, the helicase, which is 2G. So we put in helicase here, I'll stick in 2G. This is the helicase, but it's also kinase that will phosphorylate parts of the polymerase and fire it off. And now we've got our basal complex. And now transcription will occur. Now once we light this thing up, what's going to happen is the polymerase and a couple other of these basal factors like the helicase, the 2H, and one or two of the other basal proteins, those are going to move along with the polymerase, and the rest of the stuff is left behind. So we sort of end up with something a little bit more. We've got 2G that you need, that's the helicase. It's going to kind of ride in front of the polymerase, and a couple of these other guys, and then the rest of the stuff is going to be left behind on the promoter. So once the polymerase moves, it's not going to take the whole basal complex with it. It's going to take a few of the proteins that it needs, and then the rest of it stays behind on the promoter, and eventually it'll fall off. You look like you had a question, Jeremy.
Where's 2H?
2H. Where did I put that in? No. Oh, that's 2H. Sorry. This is 2H. Sorry, that's the wrong one. Wrong one. 2H. Okay, there it is. I made a mistake. It's 2H, not 2G. Okay. Sorry about that. Good point there. Okay, so now we've got our basal complex. Transcription is starting and it's continuing along. And that gives a basic idea of how this thing works. Yes?
This circle portion over here,
Uh-huh.
what is it?
That's an approximate idea of what's left behind. We leave most of these proteins behind the polymerase. As a matter of fact, we're going to take here with the polymerase. Okay, we're going to take 2F and 2H are going to come along at the minimum. And most of the rest of the stuff, so it's basically here, here, here. Those are the proteins we're going to have at the minimum. I think there may be one or two more, but most of the rest of the stuff is going to be the TBP, the 2A, the 2B, those kinds of guys we can leave behind on the promoter. They've done their job, we don't need them anymore. We definitely need the 2H, that's our helicase. We're going to need the 2E, that's going to keep the helicase running by phosphorylation and so on. So we have a little small thing that actually does the transcription. All right, well, this is probably a pretty good place to stop time-wise as well. So we're going to do that. And everybody, please be careful on the way back. I know you guys are Southerners, right? Which means you don't know how to drive in snow. Be careful. Major thing with driving in slick roads.